Great. So I am delighted to introduce our next speaker, who is Dr. Elizabeth Heller. She's an Assistant Professor of Pharmacology at University of Pennsylvania Perelman School of Medicine. And she studies epigenetic reprogramming and its role in neuropsychiatric and neurodevelopmental disease, including SYNGAP1. And she is a recent recipient of both an NIH Director's Pioneer Award in 2017, and I'm pleased, for those of you who don't know, to let you know that she also received the 2020 SynGAP Research Fund grant. So we're excited to hear about her work. And I should say she's also the aunt of Ruby, who has a SynGAP1 variant. So thanks, Liz, for being here today.
Thank you so much, Heather. Good morning, everyone. It's really my pleasure to speak to you today and to thank heartily SRF for funding this project, which is a new direction for the lab. And our role will be to elucidate some of the ways that SynGAP1 gene is regulated. So this is my niece, Ruby, at my wedding many years ago, and she's now 14. And I'll first tell you that the way that we look at gene regulation is by analyzing a feature of biology called the epigenome. The epigenome refers to the structure of DNA and the regulation of DNA based on modifications to that structure. So you're probably all somewhat familiar with chromosomes, and the chromosome is actually a really highly condensed macromolecule that consists of DNA and protein. The proteins are called histones, and both the histone proteins and the DNA that wind around the histones can be chemically modified. Those modifications can direct the proteins that then turn the genes on and off, and can also control how accessible the genes are to the transcriptional machinery. So what you can see here, these illustrations by a wonderful artist from the Penn Design School that works with our group named Juan Lee. What you can see here is that when the histone proteins are modified, the DNA can become more loosely wrapped around them, allowing for the transcriptional machinery to come in and express the gene. So when the chromatin is closed or compacted, the gene is off, and when it's open and accessible, the gene can be turned on. And so, as you know, with SynGAP1 being a haploinsufficient gene, there is in the kids a good copy. And the idea is that if we could turn up the volume or turn that good copy on to express higher, we might be able to rescue the disorder. But without understanding how the gene is naturally turned on and off, it's harder to make that tool work with maximal efficiency. So the first approach that we'll take in this new space in the lab is to look at published, publicly available data on the epigenome in both mouse and human. The first member of my lab that joined this project is Sungjun Zhu. She's now moved on, but I really wanted to share with you the response to my group when I told them we were going to go in this direction. Thanks, Liz, for the kind words. I'm just happy to contribute to something with such a meaningful purpose. Again, thanks for including me with this. Given the conservation between human and mouse on this gene, which Dr. Kennedy just showed us, we are poised to apply these tools available in our hands and potentially make meaningful, positive progress to the therapeutics. So we typically study neuropsychiatric diseases of addiction and depression, but everyone in my group was really excited to embark on this new project. So what you see here are data from other scientists that have analyzed the epigenome and have deposited that data in a public data bank for all scientists to use. And these are showing here, this is the direction of the gene, and you see the SynGAP1 gene down here and all of its different isoforms, which we just heard alluded to. And you can see this enriched signal at the beginning of the gene. And some of this signal is what tells us that this gene was available and turned on. Here we see the actual RNA of the gene. And so familiarizing ourselves with these patterns of expressions and of modifications to the epigenome can give us clues about how the gene might be regulated and how that regulation compares between mouse and human. I just wanted to point out a couple of features about regulating the gene in this way. So I show you here song annotated where Ruby's variant is in the SYNGAP1 gene. And a lot of kids with the SYNGAP1 haploinsufficiency have a variety of different variants. And so it can be difficult sometimes to envision genetic cure when there are so many different variants that need to be corrected. In this approach, we would actually target a portion of the gene that's upstream of the region that codes for protein. And that region is highly conserved and not typically mutated, or actually, I don't think there are any cases where it's mutated in a SYNGAP1 patient. So this allows us to develop tools to turn on the SYNGAP1 gene that would be applicable to all versions of this gene and potentially really valuable in that way as a therapeutic. So once we analyze the human and mouse available data, we of course then want to generate our own data. And one of the strengths of our lab is to actually analyze gene expression and the epigenome in very specific populations of neurons. So one of the key complexities of the brain is the vast number of neurons and the vast number of different types of neurons. And the neurons not only look different and behave differently, but they also express different genes, even different isoforms of the same gene. So being able to analyze specific neuronal subtypes may help us to know, and has shown in other groups as well, how the gene is regulated in the neurons that actually matter for improving function in SYNGAP1 patients. So now Songjun has moved on to a job at GSK, and Shuo Zhang has joined the lab to pick up. He is the one that will benefit most directly from the recent funds in the form of fellowship coverage. So in order to analyze specific neurons in the brain, we have to use some genetic tricks. You can't dissect out specific neurons. They're so intermingled, and their morphology, the complexity of their dendrites and axons, as well as their minuscule size, make it so that we have to use some tricks of biochemistry and genetics in order to isolate them. And roughly what we do is we take a mutant mouse that has a protein, mutant protein or fusion protein expressed only in a specific subset of neurons. And those neurons then can be isolated using an antibody that recognizes that mutant protein. We can then purify those specific nuclei from those neurons and analyze the chromatin and the RNA using some new high-tech approaches. We then use next generation sequencing to analyze it. And in a few minutes, I'll tell you about how we can then also manipulate it. So I have some really preliminary but exciting new data from some of our approaches between several members of the lab that I wanted to share with you today. And what you can see here is that when we look at a cell culture, so not from mouse brain, but a culture of glioblastoma cell line, we can see that this modification of histone, which marks active genes, is present actually at 2 locations. And this is interesting because the SYNGAP1 gene is known to have these N-terminal isoforms, whereas it may start actually at several different locations. And prior to our analyzing the DNA, the epigenome this way, we didn't have such insight into what epigenetic mechanisms might be regulating these different isoform expressions. Importantly, we also see from some published, from our published data from a very specific brain region in the striatum, also has the presence of these 2 potential start sites, as well as a specific subset of excitatory neurons from the forebrain. So it looks like there may be a pattern of expression based on epigenetic regulation of alternative start sites. Here you can see the RNA expression, which shows in the nucleus accumbens that these isoforms that start more downstream of the gene may be more abundant than those that start more upstream. And I'll show here recreation of a schematic from a paper from McMahon in 2012 that used a different method in order to analyze expression of isoforms. And as Dr. Kennedy just mentioned, there are a number of N-terminal differences in how this gene is expressed. And what McMahon showed was that beyond what we had priorly measured, even within one of these N-terminal isoforms, there can be alternative starts as well, which essentially amounts to there being an even greater number of expressed isoforms in the mouse brain. And we can now add to this body of knowledge looking in specific brain regions and specific cell types. Importantly, we can use more high throughput method called mRNA sequencing and splicing analysis, which will allow us to get the complete picture of all the splice variants expressed in the nucleus accumbens. So priorly, it was only possible to look for splice variants sort of if you knew what you were looking for. You could use degenerate primers and cloning methods to try to get all the spliced isoforms, but invariably it was difficult to get the full picture. Using deep sequencing methods and a high number of samples, in this case, we had 6 individual mouse nucleus accumbens, and we sequenced at a much greater depth. We can actually read out all of the spliced isoforms present in a given brain region. And so this is not yet schematized, it's somewhat raw data, and actually Songjun put it together this morning for me because I had a feeling isoforms was gonna come up. But what you can see is that some of the expression is of isoforms that start at this very first exon, and other isoforms are starting their expression at these downstream exons. We can also see internal alternative start sites within exons and other splice events, including some of the C-terminal events that have been previously characterized. What's interesting about this method is that it can tell us which splice variants are present, which cell types contain which splice variant, and then in combining with our epigenetic data, we can start to gain insight into what regulates those splicing events. We can also look at the spliceosome machinery, splice factors, their enrichment at specific sites, as well as the motif analysis. So the previous slide was actually the next slide. But I wanted to mention that we know that SYNGAP1 is activity, its expression can be activity dependent. And so in my lab, we study drugs of abuse, and specifically we study cocaine. And so we have a very large data set looking at mRNA expression with and without cocaine. Now, cocaine is not something that we're thinking about in the context of the SYNGAP1 patients, but it is a very robust form of neuronal activation, including synaptic activation. And so we analyze this data set as just a first pass to see if this type of neuronal activation changed the type of isoform expression. And what we can see in our very preliminary data, and it's difficult to see by eye here without the analysis, but the analysis indicates that when the neurons are activated by cocaine, this exon is skipped after actually a month of abstinence, which tells us that the first initial bout of activity can lead to a change in this SYNGAP1 expression that is persistent. So this is the type of biology that we would like to fill in using our methods. A critical advance that I pioneered in my postdoc, and that really allows us to understand the relevance of the epigenome, is epigenetic editing. So we can imagine that the modifications to the chromatin may be present because of a developmental profile, but no longer relevant to the activity-dependent expression of a gene in a grown individual or animal. Or we can imagine that the epigenetic modifications have to work in combination or in a specific context in order to evoke their effects. So the data that I showed you before, we can measure these modifications and see their relative abundance at a specific gene, but understanding that they're actually causally relevant to the expression of the gene requires a different type of experimentation. That involves testing whether or not the presence or absence of a specific modification changes the expression of the gene. And we can do that by epigenetic editing. This is a novel technology, although not so novel anymore. I now have my lab 6 years, so it's getting much more mainstream, and many, many labs use this technology in brain, in vivo, to regulate genes involved in developmental disorders, including Rett syndrome, Fragile X, and neuropsychiatric diseases, such as the genes we study. The idea is that we design a molecular tool that recognizes only the SYNGAP1 gene and contains an enzyme that can modify the histone proteins only at that gene. Then we can measure the expression of the gene and prove that that histone modification was actually causally relevant to the regulation of SynGAP. There are many different ways to edit the epigenome. The tech that I started with was called zinc finger proteins, and now we've moved on to use CRISPR tools, which many of you have learned about already. The beauty of the CRISPR approach is that it's very relatively straightforward to design because it uses an RNA recognition domain, and RNA and DNA are homologous and can be easily read out and synthesized. So what we do is design guide RNAs that recognize the SYNGAP1 gene promoter, that early stretch of the gene that regulates its expression. We synthesize these and package them into plasmids to express in the brain. We then also express in the brain the dCas9 protein, this protein component of the CRISPR package. The dCas9 is a Cas9 protein that's been mutated, so it no longer cuts DNA, which was its original function. Now what it does is bring itself, using the RNA guide, over to the gene of interest. We can fuse, synthetically fuse the dCas9 to an effector domain. And the effector domain is sort of the functional part of our tool, in that this is the enzyme that will modify the histone. So now we have these three parts, the RNA that recognizes SYNGAP1 and the dCas9 fused to an effector domain that will modify the SYNGAP1 associated histones. This is just starting in the lab now that the grant is funded, and Shuo has joined, and Shuo has just designed 5 different guide RNAs that target this upstream region of the SYNGAP1 gene. Now, I showed you before that some of the isoforms start here, but some start downstream. So we'll first work with these guides and see if we can activate expression in the brain of mice using these tools. Likely one or another will be the most effective. And then we can play around with other targeting strategies to see if we can regulate expression of different isoforms. We can fuse to our dCas9 an effector domain that will just turn on expression regardless of the chromatin context, or other effector domains that will really specifically manipulate the histones in order to see which histone modifications are most functional and effective at turning on SYNGAP1. We showed this gene image browser view to show the relevance of really understanding the context of the gene in the genome. As you can see, there's a downstream gene, cut A, that is in the opposite direction of SYNGAP1. So you can imagine if we target an activator to this region, it could also target the downstream gene, depending which gene you're looking at. And these are the kinds of specific controls that we will measure in order to make sure that our tools are completely specific. We've done this work for, I don't know, a half dozen genes now expressed in vivo to great success, both in manipulating the gene's expression in specific brain regions, as well as specific cell types, and been able to affect gene expression and behavior of animals. Just briefly, I'll mention that this approach has been used by Vania Broccoli's group, by Gaia Colasante, to reactivate the SCN1 alpha gene and rescue seizures in the Dravet syndrome. And we can see here just a similar strategy where they targeted single guide RNAs to 2 different promoters of the SCN1A gene, which is mutated in 1 copy in Dravet patients. And you can see that when they activated the gene in hippocampal neurons, they get an increased expression of the SCN1A gene. And importantly, I don't have the slide here, but the paper is published, they're able to rescue seizure behavior in these animals. There are many different ways to deliver epigenetic editing tools to the brain, as well as many different ways to regulate them. But I will pause here to allow, or to thank my group and leave time for questions. So I just wanted to point out Shuo, who has newly joined the lab and will be taking on this project as his main focus, as well as the other members of my lab and our key collaborators across Penn and other institutions. I especially like to thank our funders and the SynGAP Research Fund for inviting me today and supporting our endeavor into this field. Thank you.
Thank you. That was fantastic. Really exciting work, and it's great that SRF is helping to fund it. Let me check the question and answer box. So Janie has a question. Very exciting work. When you deliver the editing tools, how long does the histone alteration last? And what is the half-life of a particular histone in a neuron?
It's a great question, Janie. Thank you. So I'll take the second part first. So one of the reasons that neuroscientists started to get interested in the epigenome is because histones rarely turn over. They're actually some of the longest lasting cells, proteins in every cell in the body, even cells that divide. So because the brain is responsible for memory, and in the diseases that I study, chronic neuropsychiatric diseases, histone modifications were attractive as a potential mechanism that could last the lifetime of the individual. Neurons don't even divide, so the histones in the neurons are pretty much permanent. Certain histone modifications are highly persistent and others are more dynamic. The histone modifications that we manipulate, we've measured out about 2 weeks. So part of the reason that we haven't looked longer term involves our research questions and also involves the type of targeting that we use. So when we're expressing those DCAS9 tools right now, the tool itself only lasts a little while. So the expression of the DCAS9 with the plasmid expression method is not that long. It peaks at 7 days and is down by 14. The histone modification could last much, much longer, but I don't have the data to share today. There are other methods of expressing the CRISPR tools, such as an integrated virus like AAV, that they would express for the lifetime of the individual or the cell. But these are exactly the kinds of questions that we plan to pursue. And we also plan to look at how, I didn't mention this in the talk, but we want to analyze those isoform expression and the chromatin across development. So, you know, this gene is known, SYNGAP1, to sort of peak expression around postnatal day 14. And we want to fill in the regulatory mechanisms across the developmental timeline also, and then pair that with these manipulations. It may be that some manipulation is longer lasting when you put it earlier or when you put it later.
Great. Second question in the Q&A box here is, one advantage of targeting the region you do is that it works for many SynGAP variants, which is obviously great. So for a different target with a dominant negative effect, could you envision allele-specific targeting?
Thanks, Janine. This is a great question.
Yeah.
So the allele-specific targeting has definitely come up because one of the things that, in my approach, the promoter region targeted tool would target activation for both the mutated copy and the normal copy. And there is a potential disadvantage of activating expression of a mutated gene. There may not be an effect if the RNA is rapidly degraded, but we don't necessarily want to muck up the neuron with, you know, an abundance of mRNA that's destined for degradation. So there's some recent work from Bev Davidson at Penn on Huntington's disease, actually. And it seems that there are some SNPs, so some variants that segregate with the mutated Huntington, but are not toxic. So you can sort of take advantage of that allele-specific SNP to design your guide such that your guide would then be targeted to either the good copy or the bad copy. In terms of the dominant negative, as far as I and my understanding of SynGAP biology is nascent, but I think that the direction is to correct the haploinsufficiency that actually the mutated copy of SynGAP is typically rapidly degraded. So I don't know that we need a dominant negative in that context, but I certainly wouldn't rule anything out at this stage.
Okay, we'll take one last question from Janie before we move on. Can you look at expression levels of other proteins that interact with SynGAP to look at variability and severity of disease? For instance, do individuals with lower expression of SynGAP partners do better?
Yeah, it's such a great question. So this is one of the exciting things about our approach, is that we get the whole the whole transcriptome or the whole epigenome. And so we're hoping to be able to do network analysis on our animals across development to see, network analysis of mRNA is not the same as looking at protein-protein interactions, but it does tell us a bit about how SYNGAP1 is regulated in the context of other molecules. And then of course we can look for those proteins, the RNA of those proteins that we know are interacting, and analyze it just in the way that you suggested. It's a great idea. Actually, I'm gonna jot down all of this, send it to Shwo for further discussion. So thank you.
Super. Yeah, I think those modifiers of disease severity are really tough to get at, and this would be an interesting way to look at that.
Absolutely.
Thank you, Liz. That was really wonderful.
Thank you.
Keep up the good work. We're going to move on.